Hello, my name is Daniela Rodriguez, and I will be presenting Disruption of mTORC1 Rescues Neuronal Overgrowth and Synapse Function Dysregulated by P10 Loss by Tariq et al. 2022. Just to briefly outline what I'll be going through today. First, I will give a brief background on what exactly the AKT-mTOR signaling pathway is and some important proteins that are going along this pathway. Then I'll mention some important methods that they used in this paper and then get into the results. And finally, I'll highlight the main takeaway from this paper and its relevance to future studies. So to jump into the background, the AKT-mTOR signaling pathway is involved in many important functions such as protein synthesis, cell growth, and cell proliferation. This pathway includes two protein complexes that are very similar but differ in their scaffolding proteins, mTOR complex 1 or mTORC1 and mTOR complex 2 or mTORC2. mTORC1 selectively associates with Raptor, while mTORC2 selectively associates with Richter. While studies have shown that the two complexes play roles in different functions, the pathway is complex, and it does leave room for crosstalk and feedback among the effectors of these complexes that can ultimately influence their signaling. Phosphatase intensin homolog deleted on chromosome 10, or P10, is a negative regulator of this signaling pathway. Mutations in the P10 gene are found in patients with autism, epilepsy, and macrocephaly. In mouse models, P10 loss results in neuronal hypertrophy, hyperexcitability, seizures, and ASD-like behaviors. Studies show that rapamycin, a naturally occurring mTOR inhibitor, has been shown to prevent neuronal aberrations in mouse models of P10 loss. While it is thought that rapamycin inhibits hyperactivity of the AKT-mTOR pathway by inhibiting the formation of mTORC1 only, some studies have revealed that chronic treatment with rapamycin may also result in inhibition of mTORC2 activity. More interestingly, there is evidence that disruption of mTORC2 by targeting Richter can correct seizures and behavioral phenotypes associated with P10 deficiency. Because of this, it is unclear which of the P10 loss-mediated phenotypes are due specifically to mTORC1. This led researchers to their main research questions. Given the evidence that P10 loss causes overactivation of AKT-mTOR signaling and abnormal neuronal form and function, what is the precise downstream molecular mechanisms that cause these observed phenotypes? Specifically, what role does mTORC1 play in these P10 loss-mediated phenotypes? So to go over some important methods they used in this paper, they wanted to target newborn granule neurons to see the effects of knocking out P10 and Raptor with or without the presence of rapamycin on these neurons. To do this, transgenic mice were bred so that the genes of interest were floxed with LOXP sites. At P7, the mice were co-injected into the dentate gyrus with a retrovirus encoding for a fluorophore with a downstream CRE to cut at these LOXP sites, and a control retrovirus with just a fluorophore and no CRE. This technique is very useful as the effects of specific gene knockout compared to wild-type cells can be seen within the same cell population of one mouse. For the first set of experiments, after P10 knockout at P7 by injection, the mice were injected with vehicle or rapamycin from P10 to P31 daily to inhibit mTORC1, and then they had their tissues collected and imaged. The second set of experiments knocked out P10 as well as Raptor at P7, and then they had their tissues collected at P28 for imaging. Moving into the results, after knocking out P10 in the newborn granule neurons, which are red labeled cells in this figure, immunohistochemical analysis revealed that rapamycin treatment significantly rescued the greater soma size observed in vehicle-treated P10 knockout granule neurons. Also, significantly farther migration of vehicle-treated P10 knockout neurons along the granule cell layer was observed, but this was also rescued by rapamycin treatment. Additionally, rapamycin treatment rescued the increased spine density of vehicle-treated P10 knockout neurons in the middle molecular layer. Together, these results suggest that P10 loss-mediated neuronal hypertrophy can be rescued by rapamycin treatment. Next, they investigated the effect of rapamycin treatment on observed dendritic overgrowth that is mediated by P10 loss. Retrovirally infected and immunolabeled granule neurons were imaged and then reconstructed, and their dendritic arbor was analyzed. Shoal analysis revealed that vehicle-treated P10 knockout neurons had an increased number of intersections when compared with wild-type control neurons. This increase was completely rescued in rapamycin-treated P10 knockout neurons. The total dendritic length was also increased in vehicle-treated P10 knockout neurons. But again, this overgrowth was rescued with rapamycin treatment. So based off the results from the last two figures, together they suggest that rapamycin effectively rescues several morphological alterations caused by P10 loss. Because evidence has shown that chronic rapamycin treatment can inhibit mTORC1, they next investigated if this rescue of P10 knockout phenotypes observed with rapamycin treatment was mTORC1 specific by knocking out Raptor. In this figure, the neurons knockout for Raptor and/or P10 are labeled in green. They observed that P10 knockout neurons had significantly greater soma size when compared with their wild-type controls. However, this increase in soma size was completely rescued in P10 and Raptor double knockout neurons. Additionally, the P10 knockout neurons migrated significantly farther along the granule cell layer when compared with their wild-type controls. But again, P10 and Raptor double knockout completely rescued this migration. Dendritic spine density was also found to be significantly increased in P10 knockout neurons, but this increase was reduced to wild-type density numbers in P10 and Raptor double knockout neurons. Together, this data suggests that P10 loss-mediated neuronal hypertrophy can be rescued by targeting Raptor to disrupt mTORC1. They next examined the role of mTORC1 in dendritic overgrowth seen in P10 loss. Retrovirally infected immunolabeled P10 knockout granule neurons as well as P10 and Raptor double knockout granule neurons were imaged and reconstructed. Shoal analysis revealed that P10 knockout neurons had an increased number of intersections when compared with wild-type control neurons. This phenotype was completely rescued in P10 and Raptor double knockout neurons. The total dendritic length was also increased in P10 knockout neurons, which was rescued in P10 and Raptor double knockout neurons. These results suggest that mTORC1 plays an important role in the development of P10 loss-mediated dendritic overgrowth, which can be rescued by targeting Raptor to disrupt mTORC1. The rescue of P10 loss-mediated dendritic overgrowth by genetic knockout of Raptor supports the results observed by rapamycin inhibition of mTORC1 and points towards an integral role of mTORC1. The activation of mTORC1 leads to S6 kinase-mediated phosphorylation of downstream S6 protein, which is a ribosomal protein. To determine whether the knockout of Raptor leads to the disruption of mTORC1 activity, S6 phosphorylation levels were analyzed. Immunohistochemical analysis showed elevated levels of phospho-S6 in P10 knockout neurons compared to their in-tissue wild-type controls. This indicated increased mTOR activity. mTORC1 activity specifically. Interestingly, this elevated phosphorylation of S6 was completely rescued in P10 and Raptor double knockout neurons when compared to wild-type neurons. This indicates successful disruption of mTORC1 and its activity. Another crucial kinase in the mTOR pathway is kinase AKT. Knockout of P10 resulted in an increase in the phosphorylation of AKT at threonine 308. The P10 Raptor double knockout displayed a greater increase than seen in the single knockout of P10. Loss of P10 also increased the phosphorylation of the mTORC2 target, AKT serine 473, and this increase was even greater in the double knockout of P10 and Raptor. Together, these results demonstrate that P10 loss drives downstream activity of both mTORC1 as seen by phospho-S6 and mTORC2 as seen by phospho-AKT serine 473. Knockout of Raptor in conjunction with P10 results in feedback causing an increase in both phospho-AKT at threonine 308 and phospho-AKT at serine 473. This study found which of the P10 loss-driven aberrations in neuronal form and function are orchestrated by mTOR complex 1, or mTORC1. The data demonstrates that activation of mTORC1 is necessary to drive increased neuronal migration, somal hypertrophy, dendrite growth and branching, and dendrite spine formation that results from P10 loss. This study is relevant as P10 loss-mediated aberrations in neuronal form and function are implicated in neurological disorders like ASD and epilepsy. And this is relevant because some of the epilepsy cases that are currently ongoing are resistant to current treatments. So understanding the mechanisms that underlie this are crucial in finding effective treatment for these patients So more investigation is needed. Thank you for listening.